Hello students. Today we'll be discussing regarding the topic fermentative metabolism. I am Shaista from Department of Studies in Microbiology, KSOU. In today's topic, we'll be dealing with what is fermentation, the different types of fermentation, alcohol fermentation, lactic fermentation, mixed fermentation, and in lactic fermentation, homolactic fermentation, and heterofermentation. Heterolactic fermentation will be discussed in detail in today's class. Let's begin our discussion with the topic fermentation. So, fermentation is an anaerobic process. It is an enzyme catalyzed metabolic process where microorganism converts starch or sugar to alcohol or an acid and simultaneously release the energy. Fermentation is an anaerobic redox process in which the oxidation of substrate is coupled to the reduction of another substrate. There is always a difference in redox potential of the substrate and the end product, providing the energy for the ATP synthesis. The oxidation reaction is usually coupled with the substrate level phosphorylation. The reduction reaction is not associated with substrate level phosphorylation. These are some of the basic important points by which a fermentation process can be defined as. A fermentation process is the one which releases energy from sugars or other organic molecules such as amino acids, organic acids, purines, and pyrimidines. This process does not require oxygen, but sometimes can also occur in presence of oxygen as well. This process does not require the use of the Krebs cycle or the electron transport chain. This process uses an organic molecule as a final electron acceptor. This process only produces a small amount of ATP when compared to the other aerobic processes, because much of the original energy is in glucose remains in the chemical bonds of the organic end products such as the lactic acid or the ethanol. Therefore, only very small amounts of ATP is produced as much energy is conserved in the chemical bonds of the end products. So students, now let's discuss the process of fermentation. Fermentation, as we all know, is an anaerobic biochemical process. In fermentation, the first process is same as the cellular respiration. That is our glycolysis, which results in the formation of pyruvate and generation of net 2 ATP molecules during the glycolysis. This we have discussed in the earlier classes which we had taken. Now, in next additional extra step, what is happening in fermentation process is that this pyruvate is reduced to lactic acid or ethanol or other products, where NADH is oxidized back to NAD. And this is NAD which is formed is reutilized back in the glycolysis process. So this is a brief explanation of how fermentation is occurring. Now we can see in the diagram here, the glucose enters the glycolysis cycle where two pyruvates are formed from single glucose. Now, if oxygen is present, it enters the mitochondria. The pyruvate enters the mitochondria where it is converted into 2-acetyl-CoA. Two pyruvates are converted into 2-acetyl-CoA because of the PDH complex, which we had discussed earlier in the Krebs cycle or the TCA cycle. The first step where the pyruvate is getting decarbonized into 2-acetyl-CoA by the PDH complex, and this acetyl-CoA after cleavage enters the Krebs cycle and then the electron transport chain. This is a routine aerobic respiration. But what happens when there is absence of oxygen? When there is absence of oxygen, these products undergo conversion. Pyruvate undergoes the conversion into either ethanol or lactic acid or any other product, and thus resulting in the fermentation. So the absence, that is anaerobic process, is one of the important factors for the fermentation. So students, so far during fermentation, we have understood that electrons are transferred from reduced coenzymes, that is our NADH or NADPH, to pyruvic acid or pyruvates and its derivatives. Those final electron acceptors are reduced to the end products. As the essential function of the second stage is that of it must give a steady supply of NAD plus and NADP plus so that the glycolysis can keep on continuing and it shouldn't stop. In fermentation, ATP is generated only during glycolysis. That is, once our end product pyruvate is formed, there is no generation of ATP. The glycolysis itself results in the formation of ATP. Microorganisms can ferment various substrates, that is, various end products, depending on the particular type of microorganism in which fermentation is taking place, the substrate involved, and the enzymes that are present and active. Chemical analysis of these end products are very useful in identification of various microorganisms. So generally, students, we can divide the fermentation into two parts or into two pathways. One is the alcoholic fermentation and the lactate fermentation. Now, what's happening in the alcoholic fermentation? The alcoholic fermentation, the pyruvate is split into acetaldehyde and carbon dioxide. And further, this acetaldehyde receives the electrons from NADH and forms the ethanol where NADH is oxidized to NAD. This NAD again goes and can be utilized into the glycolysis cycle. But in lactate fermentation, what is happening? The pyruvate itself receives the electron from NADH, converting the NADH into its oxidized form, that is NAD, and forming the lactate. Okay, so there is no release of carbon dioxide here. So let's see here in the flowchart what actually is happening. The glycolysis, still the formation of pyruvic acid or the pyruvate is similar for both the pathways. And here itself there is a generation of the ATP. After pyruvic acid or pyruvate is formed, there is no generation of ATP. Once this common product pyruvate or pyruvic acid is achieved, it can either result in the formation of lactic acid, where the pyruvate itself accepts the hydrogen from the NADH, oxidizes the NADH to NAD, and results in the formation of lactic acid. Okay, and this NAD again can be used into the glycolysis cycle. This formation of lactic cycle is called as a lactate fermentation. But in the alcoholic fermentation or alcohol fermentation, the pyruvate is split into acetaldehyde and carbon dioxide. Now, the acetaldehyde which is formed accepts the electron from NADH, converting it into its oxidized form NAD. After the acetaldehyde accepts the hydrogen from NADH, it results into the formation of ethanol. When the pyruvic acid is split into the acetaldehyde and carbon dioxide, the carbon dioxide is released and the oxidized form of NAD is also received. This NAD can again be utilized into the glycolysis process. Thus, the major two different pathways. One is the alcoholic fermentation pathway, the another is the lactic acid fermentation pathway. So students, now let's brief of the summary of this fermentative pathway process. So we have understood that glycolysis is the first step of the fermentation pathway, where from one glucose molecule, two pyruvates are formed, two NADH are released, and two net ATP are gained. This is from our glycolysis process. But after glycolysis, what is happening is that the pyruvate is converted into other molecules, but this pyruvate is not fully broken down into carbon dioxide or water. Okay, it is not fully broken down into carbon dioxide and water, but it is converted into some other molecules in absence of oxygen, and thus it regenerates NAD but does not produce an ATP. Okay, this we have understood, and I have earlier explained this point as well. Okay, provides enough energy for okay, now whatever the end products are formed, okay, and it provides enough energy for some single cell or an anaerobic species to survive. Okay, whatever energy is there, it may produce less energy when compared to the aerobic cycle, but because most of the energy is stored in the chemical bonds in the end product of the fermentative pathway, but whatever energy is produced by this process is sufficient enough for some single cell and anaerobic species to survive. So now, students, we had discussed earlier that based on the microorganisms in which the fermentation is occurring, the substrate which is involved, the enzymes that are active and present, the end product of the fermentation can differ. Therefore, the end product can be alcohol, or an lactic acid, or an acetic acid, or an alkaline products resulting in alkaline fermentation. And this type of alcoholic fermentation occurs mostly in yeast. Lactic acid fermentation occurs mostly in lactic acid bacteria. Acetic acid fermentation occurs in acetic acid bacteria, and alkaline fermentation occurs mainly in bacillus species. These have their own specific utilization, like alcoholic beverages, or the lactic acid production, or the acetic acid production, or the amino acid production. These all is bifurcated mainly depending on the type of microorganism that is involved, the types of substrate that are involved, and the types of enzymes that are active and operating. Based on this, the end products of the fermentation can be divided in vast variety, and thus their application as well. So students, now let me generalize the different types of fermentation. So the first type of fermentation, depending on how many products are being formed, how many types of products are being formed, uh, it can be classified as homofermentation. That is where only one end type of product is being formed. Or a heterofermentation, where more than one type of product is being formed in the end product. That means two products can be formed as end product, or the three products can be formed as an end product in heterofermentation. But whereas in homofermentation, only one end product is formed. Okay. So now on the basis of the end products form, fermentation can be classified as follows. So the end product nature, what kind of product is it? Is it an alcohol or an acid, or it's a mixture of both? Based on that, now the based on the nature of the end product, the fermentation is classified as the lactic acid fermentation, or the alcohol fermentation, or mixed acid fermentation. Okay, now we'll be studying all this fermentation in detail. Okay. Now let's discuss our first type of fermentation, that is alcohol fermentation. As similar to the lactate fermentation, first glucose need to be converted into pyruvate by the action of glycolysis process. Then this pyruvate is converted into ethanol by action of two enzymes. First enzyme is that pyruvate decarboxylase, and our second enzyme is alcohol dehydrogenase. The, the first step of this process is that the pyruvate undergoes decarboxylation to form acetaldehyde by release of carbon dioxide during decarboxylation. Pyruvate is 3-carbon compound, okay? There is a resulting release of carbon dioxide, so it is converted into 2-carbon compound due to the process of decarboxylation by the enzyme pyruvate decarboxylase. Once acetaldehyde is formed, it is converted into ethanol by the action of the enzyme alcohol dehydrogenase. Now what's happening? The acetaldehyde accepts an extra pair of hydrogen ion from NADH, reducing the NADH into its oxidized form and converting itself into an ethanol. In this process, the NADH in its oxidized form is generated, which is again utilized into the glycolysis process. Okay, so now let's look at the equation. So glucose with 2 ADP. This ADP is used up in the glycolysis process, okay, where we have again net generation of 2 ATP coming from the glycolysis process, okay. After pyruvate is made, or there is no production of ATP. This we have to understand. But when we are including the glycolysis in alcoholic fermentation, this generalized equation is used. That is, glucose, adenosine diphosphate, and phosphate results in the formation of 2-ethanol. From one molecule of glucose, we are getting 2-ethanol, 2-carbon dioxide, and 2 ATPs. These 2 ATPs are nothing but the net ATPs which are generated in the glycolysis process. Okay, now which examples can we give to alcoholic fermentation? It can be seen in the Pseudomonas species or a Saccharomyces cerevisiae species. Okay, so students, now let's discuss about the second type, that is lactic acid fermentation. So once our pyruvate is formed by various metabolism, the pyruvate is converted into lactic acid just by a single step, which is carried out by the lactic acid bacteria. So based on the end products, the lactic acid fermentation can be divided into two types, that is homolactic fermentation and heterolactic fermentation. Now let's look at the homolactic fermentation. Where do you find this flowchart, this pathway? Can you identify this pathway? What is happening here? This is nothing but our glycolysis. Our glycolysis pathway, where the glucose is converted into pyruvate. And now what happens? This pyruvate accepts one of the hydrogens from the NADH, where NADH is reduced to NAD in the presence of an enzyme called pyruvate dehydrogenase, or a similar enzyme which converts the pyruvate into the lactate. Okay, so by accepting an hydrogen from NADH, pyruvate is getting converted into lactate, and the reduced form of NAD is again utilized into the glycolysis process. This is the homolactic pathway, where only a single end product lactate is formed. But whereas in heterolactic pathway, you can see which cycle is this, which pathway is this, by which we have already discussed in this in previous other classes. This pathway is nothing but our pentose phosphate pathway, where ribulose or pentose is formed. Okay, our end product ribulose is formed. This ribulose undergo formation of glyceraldehyde 3-phosphate because of the action of various enzymes like transketolase and transaldolase. Here series of reactions are there, and our glyceraldehyde is formed, glyceraldehyde 3-phosphate, and again from here glycolysis goes on, and the pyruvate is formed, and from the pyruvate the lactate is formed similarly. But simultaneously there is a production of acetic acid, where the acetic acid is converted into ethanol by formation of an intermediate called acetaldehyde, which accepts the hydrogen electron from NADH, again getting reduced form into NAD and formation of ethanol and carbon dioxide. Here decarboxylation is also occurring. Therefore, there are three products here. One is lactate, the second is ethanol, and the third is carbon dioxide. Thus, a heterolactic pathway where three products are formed, and homolactic pathway only one lactate is formed. Okay, now we will discuss each one in detail. So students, now let's talk about the basic points of homolactic fermentation. So homolactic fermentation is the end product is only one product, that is our lactic acid, where the pyruvate is reduced to the lactic acid or the lactate by the action of enzyme lactate dehydrogenase or pyruvate reductase. Okay, where can we see this occurring? We can see this occurring in some of the species like Streptococcus thermophilus, Streptococcus lactis, Lactobacillus lactis, Lactobacillus bulgaris, Pediococcus, and Enterococcus. These are the few examples which we can give for homolactic fermentation. And what can their application be? They can be used in dairy industry for storing of milk to create various fermentative products. These also help in digestive tract of humans, in which digestion of lactose present in the milk is possible. They are used as a probiotic. These bacteria are responsible for the dental caries, which is also an homolactic fermenting bacteria as well. So these are the some basic points about homolactic fermentation. Now let's see the next one. So students, by our earlier prior discussion that heterolactic fermentation, we have three end products, that is ethanol, carbon dioxide and lactate. So what's the difference between homolactic fermentation and heterolactic fermentation is that homolactic fermentation follows the glycolysis pathway, but in heterolactic fermentation, the glucose is converted into pyruvate and acetic acid and carbon dioxide by utilizing the pentose phosphate pathway, which is an alternative to glycolysis, and it is functioning parallel to the glycolysis itself. Once the pyruvate is formed, it is reduced to the lactic acid. But another product which is formed is that acetic acid is also reduced to formation of ethanol and carbon dioxide. So our end products in this heterolactic fermentation are lactic acid, ethanol and carbon dioxide. Okay, I hope the major difference over here and the process and the pathway which they are following are bit clear. Students, now let us do a comparative study of homolactic fermentation and heterolactic fermentation. By definition, homolactic fermentation is conversion of one glucose molecules into two lactic acid molecules. But whereas heterolactic fermentation is conversion of one glucose molecule into one lactic acid molecule plus one carbon dioxide plus one ethanol molecule. By process, homolactic fermentation results in the conversion of glucose into lactic acid. But the heterolactic fermentation results in the conversion of glucose into lactic acid, ethanol, and carbon dioxide. By end product, homolactic fermentation has lactic acid and ATP as end products. This ATP is the one which is generated during the glycolysis pathway, which homolactic fermentation used before the production of pyruvate. By the definition of lactic acid fermentation, the end products here are the lactic acid, carbon dioxide, ethanol, and ATP. The ATP production in homolactic fermentation is high because it follows the glycolysis pathway to generate the pyruvate. But the heterolactic fermentation, the ATP production is low because it follows the pentose phosphate pathway to generate the pyruvate. Therefore, the production of the ATP is low in heterolactic fermentation. I hope now you are clear with this two type of lactic acid fermentation. Okay. Now let's go to the another point. So students, now we are discussing the last type of fermentation, that is mixed acid fermentation. In mixed acid fermentation, mixture of various acids such as lactic acid, acetic acid, ethanol, formic acid, etc. are formed. At first, at first, the pyruvate, which is generated because of the glycolysis, is cleaved into acetyl-CoA and formic acid. Because of the action of enzyme, pyruvate formate lies, which is cleaving it into the acetyl-CoA and formic acid. From formic acid, various other end products like acetic acid, lactic acid, ethanol, succinic acid, carbon dioxide, and water can be formed. And this depends on the type of pathway the various types of bacteria is utilizing. Okay. However, the formic acid that is formed in such pathway is always an intermediate product. This pathway can be followed by the members of Enterobacteriaceae family, such as E. coli and Salmonella and etc. So here the end products are as seen can be a lactic acid or an acetic acid or an ethanol, a carbon dioxide, formic acid, succinic acid, and various depending on the type of microorganism that is involved and the type of pathway this microorganism is utilizing. Thus, we end our topic fermentation, where we have discussed various types of fermentations and their pathways, and we have even discussed about the mixed fermentation as well. Okay, thank thank you, students. By this, we end our fermentation topic. Thank you.